Good afternoon, class. So today we will be discussing the chapter 3 notes, and chapter 3 deals with cells, the living units, the functional units of life. And chapter 3 is a relatively long chapter. There's a lot of material in chapter 3. Hopefully some of this material is just a review for you. Alright, so again, the cell is the functional unit of life, okay? So based on the cell theory, okay, how an organism function is going to depend on the individual and combined activities of all its cells, okay? And you remember from chapter 1, I believe, you have cells, a functional unit of life. Then a group of cells make up a tissue, all right? And then tissues make up organs, okay? And then organs make up organ systems, and then organ systems make up an organism, okay? And all the cells involved, the tissues involved, the organs involved have to work together, all right, in order for the organism to be functional and maintain homeostasis, all right. And also, based on the cell theory, just understand that cells can only arise from other pre-existing cells, okay. And with everything in anatomy, okay, we talk about anatomy and physiology, all right, structure and function are complementary. So essentially, something that you want, a cell that's going to be used for diffusion, for something to pass through its cell membrane by diffusion, you're not going to want, very thick cells. You're going to want something that's flat, all right, and that's going to make it easier for diffusion to occur. All right, so structure and function are going to be complementary, okay? Just understand that. So this is showing you that there are a lot of diversity when it comes to cells, okay? All right, there's over 200 different types of human cells, and a lot of diversity. Like erythrocyte is a blood cell, and red blood cells essentially are just bags of hemoglobin, okay? They're packed full of hemoglobin. They don't even have a nucleus, okay? So red blood cells, a mature red blood cell is just packed full of hemoglobin. That's what its function is, to transport, oxygen to other areas in the body, okay? To tissues and other areas in the body. So in order to do that, it's just packed full of hemoglobin. And then you have other cells like macrophages, okay? Nerve cells, smooth cells, skeletal muscle cells, all right, epithelial cells, okay? So again, there are a lot of different sizes, shapes, subcellular components, okay, that lead to different functions, all right? So all these cells have different functions. All right, but in general, we're going to just talk about the general structures that all cells have to have, at least these 3 basic parts, and then they can have some variety when it comes to other organelles within the cell. But you got to have a plasma membrane. So remember, the plasma membrane is going to be what determines what comes in and out of the cell. All right, so you have to have a plasma membrane, then the cytoplasm, okay, so that's the jelly-like fluid within the cell, the cytoplasm, okay, and then the nucleus. All right, the nucleus, you think of the nucleus as a control center of the cell. All right, the nucleus is going to be where the DNA is located, and it's, as you know, DNA is very, very important. And since the DNA is so important, okay, the DNA never actually leaves the nucleus, okay? So the DNA has, of course, the information in it that makes us, what determines our eye color and hair color and so forth. But DNA also has the blueprints for making proteins, okay? And that's very important because remember, enzymes are proteins, and cells need a lot of enzymes to carry out chemical reactions within the cell. All right. So the DNA has a blueprint for making proteins, but as we talk about this later on, in the lecture, okay, the DNA never actually leaves the nucleus. Okay, something else called messenger RNA is going to go in the nucleus. Messenger RNA is going to get a copy of the messenger RNA that's going to be correspondent to a segment of DNA, and then that messenger RNA that's what leaves the nucleus and goes to a ribosome, and ribosomes are where protein synthesis takes place. When you think of ribosomes, think of ribs. When you think of ribs, think of meat. Think of meat, think of protein. Okay, so ribosomes, ribs, protein. Ribosomes are where protein synthesis takes place. All right, so, but we'll talk about that in more detail a little bit later on. All right, so this is just an overview of the chapter. So again, we are talking about cells, okay, but we're going to be exploring the plasma membrane, the structure of the plasma membrane. We're going to talk about passive versus active transport. All right, we're also going to talk about the cytoplasm. All right, we're going to talk about the nucleus. All right, again, the nucleus houses the DNA, but remember, the DNA never leaves the nucleus because you want to keep the DNA protected. Okay, so then in order for a cell to grow and divide, we're going to talk about transcription and translation. All right, and also talk about DNA replication and mitosis. Okay. All right, so here's a diagram that has, a picture of all the different organelles labeled, okay, in the cell. And I remember, when you first, start studying biology and you see all this stuff going on and you're like, wow, this is a lot of stuff. But if you can kind of look at the big picture of what has to happen within the cell. So again, here is your nucleus, okay, control center of the cell, and a lot of students don't know what this right here is. This dark spot within the nucleus, okay, is the nucleolus. Okay, so this dark spot within the nucleus is the nucleolus, and that's where ribosomal RNA is made. Okay, ribosomal RNA is made in a nucleolus, and you essentially need that rRNA for assembling ribosomes. Okay, but that's made in the nucleolus. Okay, and then chromatin is just DNA wrapped around proteins. Essentially within a nucleus, if you didn't essentially wrap DNA around protein and DNA was just able to be free, it will take up, the entire cell, okay? Because DNA is very stretchy, and if you didn't wrap it around something, it would just take up the entire cell. So chromatin is just DNA wrapped around protein, wrapped around the histones, and essentially that's going to be useful to really keeping the DNA organized. When a certain part of the DNA needs to be replicated, the chromatin to unwind itself, and also when a certain part of the DNA needs to be copied for protein synthesis, needs to be copied into messenger RNA, the chromatin will also unwind itself, okay? But then once it's finished doing what it's doing, it'll wind itself back up, okay? So that keeps the DNA nice and condensed within the nucleus, okay? And then you have a nuclear envelope. So the nuclear envelope, just think of it as a membrane that surrounds the nucleus. All right. And then, so let's just talk about, let's work from the inside of the cell out, okay? So we have the nucleus, and you see you have these nuclear pores. You see these nuclear pores right here, channel pores right here in the nucleus, and then you also see right continuing with the nuclear envelope, with the nuclear membrane, nuclear envelope, continuous with it is the endoplasmic reticulum. Okay, so you see this is the smooth ER, smooth because it does not contain ribosomes. So that's a smooth endoplasmic reticulum. All right, but then you also have what is called the rough endoplasmic reticulum, and it's rough because of its appearance, and the reason why it looks rough is because it has ribosomes attached to it. Okay, so again, I'm gonna say this again, continuous with the nuclear envelope, the nuclear membrane, is the endoplasmic reticulum. It's continuous with the nuclear membrane. And you have two types of endoplasmic reticulum, smooth ER, and the smooth ER is where you're going to have your lipids are going to be made. Lipids are going to be made in the smooth ER. And the rough ER, rough endoplasmic reticulum, is where you're going to have your proteins are going to be synthesized. Now, here's what you have to understand, okay? Ribosomes are where proteins are synthesized, okay? So the ribosomes in this instance are attached to the rough ER. So the ribosomes are attached. So it's still, the proteins are still being synthesized on the ribosomes, but this ribosome attached to the rough ER. But you also have, just understand too, you also have ribosomes that are free, not attached to anything within the cell. So you see right here, you have free ribosomes that are not attached to anything within the cell. All right, but the reason why ribosomes attached to the rough ER is significant because, again, working from inside out, what happens is proteins, a lot of times you have proteins that are going to be destined to stay within the cell. Okay, if the protein is going to be destined to stay within the cell, a lot of times what happens is it's going to be made on a free ribosome that's just in the cytoplasm, not attached to anything. But proteins that are destined to go to different locations, different organelles, or to be secreted outside the cell. A lot of proteins that cells make are excreted, secreted outside of the cell. So what happens is it goes from the rough ER, so the proteins made on a ribosome on the rough ER. From the rough ER, think of the rough ER as kind of like a highway, okay? A highway that's taking you to the next location, and the next location from the rough ER, the highway is taking you is to the Golgi apparatus, okay? And right here, you see the rough ER takes you to the Golgi apparatus, and what happens at the Golgi apparatus, think of the Golgi apparatus as the post office of the cell, okay? So at the post office, remember, things get sorted and packaged to go to a different location. So the Golgi apparatus is going to take the proteins and also the lipids from the smooth ER. So the lipids from the smooth ER also go to the Golgi. And then what happens is the Golgi apparatus takes the proteins or the lipids and package it, and then it sends it to where it needs to go, okay? So if it needs to go to a lysosome within the cell, it's going to leave the Golgi apparatus and it's going to go to that lysosome. If it's going to be destined to be in the plasma membrane of the cell, it's going to go from the Golgi apparatus. You see these little vesicles, those little circular vesicles that are filled with protein. It's going to go right here to the plasma membrane. And then if it's destined for the plasma membrane, it's going to stay in the plasma membrane. And a lot of the lipids that you make within the cell, a lot of the lipids that you make in the smooth ER are going to be destined for the plasma membrane. It's going to stay there, okay? But if it's something that's destined to be secreted outside of the cell, what happens is this vesicle fuses with the plasma membrane, and then it goes through what's called exocytosis. Okay, exocytosis is when something, think of the cell kind of spitting out the protein. Okay, it goes through exocytosis, and it's going to release the vesicle is going to fuse with the plasma membrane. It's going to release the proteins outside of the cell. Okay, exocytosis. All right, so that should let you understand what the smooth and the rough ER are for, and the Golgi apparatus. Okay, so that's took up a big chunk of what's going on within the cell. Okay, and then the other stuff, right here, you have the mitochondria. Okay, mitochondria is an organelle that's used to make ATP, and ATP is the energy currency of the cell. Okay, and the name organelle. Okay, so you have these different organelles within the cells. Okay, and the name organelles comes from, it means little organs. So you think about the organs that you have in your body. For instance, you have kidneys, hearts, lungs. All those are different organs. Okay, and they all work together so that your body can function. Okay, so it's similar. That's why they call them organelles, little organs or little bodies, because they're similar to how the organs in your body, okay, have to all work together for your body to function. All the organelles and the cell have to work together so that the cell can function. Okay, organelles, little bodies, little organs. Okay, so think of the analogy with the organs in your body, how they all have to work together for your body to function. All these organelles within the cell have to work together for the cell to function. All right, so you have the mitochondria makes the energy. All right, so that would be like the generator of the cell. If the cell was a factory, okay, that would be like the generator of the cell. All right, so then again, the plasma membrane is what keeps things going in or out. All right, it's semi-permeable, meaning it lets certain things in and out, but only certain things. And we're going to talk more about the structure of the plasma membrane later on also. Okay. So you have these centrioles. Okay, so these centrioles are really important when it comes to mitosis. So, you know, during mitosis, your chromosomes have to get moved around. All right, your chromosomes get moved around from the middle of the cell, then they get separated, and so forth and so on. So they get moved around during mitosis. Okay, so these centrioles are what make those microtubules that attach to the chromosomes during mitosis, and it moves them around. Okay, so that's what those centrioles do. Okay, they're very important for mitosis. So those mitotic spindles that move around the chromosome during mitosis, okay, they're made by the centrioles. All right, so then you also have the cytoskeleton elements. All right, microtubules are the largest, and then you have intermediate filaments, which are, like the name tells you, in between. And then you have, I'm trying to see if they have it in the image. I don't see it in the image, but you have a smaller filament called the microfilament, okay, that are smaller than the intermediate filaments, okay. But the cytoskeleton tells you, the name pretty much tells you what it does. It essentially helps reinforce the cell, essentially. All right, cytoskeleton, you think of your skeletal system in your body, it reinforces your bones, you know, allow movement to occur, reinforces your body and protects certain things. Cytoskeleton of the cell reinforces these cell membrane, okay, helps keep its shape. All right, so just by talking about that, I've almost covered all of these organelles. And then you have lysosomes. Lysosomes, okay, when you think of lysosomes, think of Lysol. When you think of Lysol, you think of cleaning, right? Lysosomes are the garbage disposals of the cell. So they're going to degrade anything that needs to be degraded within the cell. It goes to the lysosome, and the lysosomes are going to degrade anything that needs to be broken down. All right, they are the garbage disposals of the cell. All right, then we already talked about the cytoplasm, the jelly-like fluid within the cell. And then you have peroxisomes right here, okay? So peroxisomes are involved in lipid metabolism. So they're involved in breaking down lipids, breaking down fats for energy. And also peroxisomes take reactive oxygen species like hydrogen peroxide, and they break them down into safer molecules like water and oxygen. Okay, and we'll talk about this some too. But one of the byproducts, so you know we all need oxygen. Our body needs oxygen to survive. And essentially, the cell uses oxygen for cellular respiration. The mitochondria uses it for cellular respiration. And it generates the majority of the ATP in the body through cellular respiration. Okay, but one of the byproducts of oxygen or using oxygen is oxygen radicals, and oxygen radicals are very reactive and very destructive. So if you don't break down those oxygen-free radicals, it's going to essentially degrade and destroy the cell. Okay, so peroxisomes are able to break down hydrogen peroxide, which is one of the byproducts of using oxygen, hydrogen peroxide into water and oxygen. Okay, so water and oxygen is not going to hurt the cell. Okay, all right. So again, cells have an inside and an outside. All right, that's going to be determined by the cell membrane. Okay, the plasma membrane separates the inside of the cell from the outside of the cell. Okay. So you have a variety of substances found outside the cell. That's called extracellular fluid. So interstitial fluid. So remember, well, we haven't talked about this. You actually don't talk about blood vessels until anatomy 2. But essentially, you go from, your arteries leave your heart, and then you go, your arteries get smaller and smaller until they become, capillaries. And then the capillaries on the other side are going to become veins. But essentially what happens at these capillaries, which are very, very thin, all right, it's a lot of fluid, well, not a lot of fluid, but it's easy for fluid to get pushed out of especially if it's a lot of high pressure, to push out of the capillaries, okay, because they're very thin, and that fluid that gets pushed outside of the capillaries becomes what's called, interstitial fluid, okay. And that interstitial fluid is going to end up being returned to the, return in the lymph. Essentially goes into the lymph, and then that lymph, passage that's going to return it to the, the veins of the heart, so it can return to your body. Okay, so it goes so there's a way that it gets essentially returned to the body, but just understand that that fluid that escapes from the capillaries, it's interstitial fluid, and then that becomes, part of the lymph that's going to be returned to the body, okay, after it goes through the lymph, passageway, and then it's going to return to one of your major veins in your body, okay? And then you have cerebrospinal fluid, okay? We'll talk about that during the nervous system. Then you have cellular secretions, saliva, mucus, and then you have the extracellular matrix, which acts as the glue to hold cells together. All right, so again, the plasma membrane, very, very important. It acts as a barrier separating the intercellular fluid from the extracellular fluid. All right, so again, it plays a dynamic role in cellular activity by controlling what enters and leaves. You cannot have a functional cell if you cannot control what enters and leaves, okay? All right, so the plasma membrane is what's called a lipid bilayer. And when you see a picture of it, you'll understand why it's called a lipid bilayer, okay? And I'll talk more about the lipid bilayer when we get to the picture because it's just easier to explain when you can see it. But then within the lipid bilayer, you can have specialized membrane proteins, okay? And these membrane proteins may be channels for certain ions or for certain substances to pass through. We're going to talk about, again, the lipid bilayer in a second, but essentially the lipid bilayer has a hydrophobic core. So the middle of it is hydrophobic, meaning it does not like water. So anything that has a charge, all right, anything that's hydrophilic, it can't just go through, diffuse through the plasma membrane. It needs these protein channels so that it can get through, okay? And then you have also sugars attached to the plasma membrane. You also have membrane structures that help to hold the cells together through cell junctions, okay? All right, so with this picture of a plasma membrane, so you see bi means two, so bilayer, okay? So you see how you got a lipids right here, and they're called phospholipids, okay, because they have a phosphate attached to the head, which makes it hydrophilic. So these are the heads, these circular phosphate attached to the head. And then the lipid tails. You see how the lipid tails point in the middle? And then it's called a bilayer because you have the same thing on this side. The phosphate head, and then you have this lipid tail. Okay, so you have those lipid tails that point to the middle of the lipid bilayer, and those lipid tails keep anything that's positively charged from passing through, all right, because it's going to repel anything that's charged from going through, okay? But and also it's not going to let any water go through. It's also going to repel any water also. But these phosphate heads, okay, remember phosphate has a charge to it, so anything charged, water is going to like, okay? So the phosphate heads are hydrophilic. They like water. So this part of the lipid bilayer phosphate head is interacting with the extracellular fluid, and this part of the phosphate head is interacting with the intracellular fluid of the cell, okay? All right, so then you see these channel proteins that are embedded in the plasma membrane. And again, that allows very large substances and charged substances to pass through the plasma membrane, okay? And then you have cholesterol right here in the plasma membrane. And I know cholesterol gets a really, really bad rep because we eat too much red meat and we get too much cholesterol. But cholesterol does serve a purpose. So this lipid bilayer is fluid, okay? So it has movement to it. But the thing is, you don't want it to be too fluid. You don't want it essentially to be moving too much, and you don't want it to be too stiff either, okay? So you don't want to be too loose or too stiff. So what happens is you have these cholesterol. Cholesterol, it pretty much embeds itself in the plasma membrane, like right here. And then what happens is it pushes those cholesterol. It's pushing when it embeds itself like that. It's pushing against the sides of those lipid tails, and essentially what's happening is it's making the plasma membrane more firm, okay? So again, you don't want the plasma membrane to be too loose. So the cholesterol plays a very important role of making the plasma membrane more firm. But again, too much cholesterol is bad for you. But in general, too much of anything is bad for you. Just remember that too much of anything is bad for you. All right, but cholesterol does play an important role in the body. Okay, but again, too much of it is bad for you. And then when these proteins, when they're embedded in the plasma membrane, they're called integral proteins. Okay, and then you have what's called peripheral proteins that are not embedded within the plasma membrane. They are just attached on the outside of the plasma membrane. Those are peripheral proteins, okay, attached on the outside of the plasma membrane. So some of the functions that this image is telling you about the plasma membrane: mechanical barrier, okay, again separates the inside of the cell from the outside of the cell. It's selectively permeable, so certain things, like small things like gases, can diffuse through. Certain hormones that are lipids, okay, so that your hormones that are lipid derived can go through the plasma membrane, okay? So certain things can go through the plasma membrane, but then again, like I said, a lot of things cannot, all right? Because you have this separation from the inside and outside, you can make what this electrochemical gradient, okay? Which is important when we get to the central nervous system and neurons. All right, the action potentials, electrochemical gradient is very important. Communications, and then cell signaling. Okay. Again, you have a lot of you see these carbohydrates, these sugars attached to the outside of the cell. Okay. Then you can also have peripheral proteins that can act as receptors on the outside of the cell. Okay. All right, so again, membrane proteins, two types: integral proteins, which are embedded within the plasma membrane, and then peripheral proteins, which are associated with the outside of the cell membrane. All right, so these are some of the different functions of the cell membrane proteins. Again, transport. A lot of things that are polar, very large, they cannot just go through the cell membrane, okay, because of that hydrophobic core. So they have these protein channels which are used for transport. You also have receptors, okay, that can take a signal on the outside of the cell and then transfer it to the inside of the cell, all right, and then that signal can be passed on to something else until it does its end result of what it needs to do, okay? So receptors for signal transduction are going to also be cell membrane proteins. And then you have attachments to like a cytoskeleton, okay, that helps give the cell membrane extra support. All right, so then a lot of enzyme activity occurs in these membrane proteins. A lot of times they have to pass a cell signal on, maybe from a receptor, pass it on to something within the cell, and then that a lot of times it's going to be through enzyme activity. It's going to be occurring. And then you have these intercellular joining, all right here, okay? So membrane proteins of adjacent cells may be hooked together, okay, in intercellular junctions. And then you have cell-to-cell recognition. All right, so some glycoproteins, so glyco sugar, and then proteins. So proteins which sugars attached. That's what a glycoprotein is. Serve as identification tags that are specifically recognized by other cells. And these same glycoproteins also are recognized by things like viruses and bacteria that make us sick too. They use these same glycoproteins like that our body normally uses for cell-to-cell recognition. They kind of hijack it, and they can use it to get themselves to get taken within the cell and then make us sick. Okay, viruses and bacteria kind of hijack that cell-to-cell recognition mechanism. All right, so again, plasma membranes are selectively permeable. All that means is that some things can come in, and then some things cannot. Okay, some things can pass through, and some things do not. Selectively permeable. All right, so you got two ways that a substance can cross the membrane. You have passive transport and active transport. And again, the name tells you whether it requires energy or not. Passive is what it says. Passive is not going to require energy. Active transport, it will require energy. Okay. So for passive transport, which does not require energy, you have two types. Okay. Diffusion. Okay. So all diffusion is something, particles moving. Some particles, some substance moving from where it's highly concentrated to where it's low concentrated. And diffusion always moves from high to low. The particles always move from high to low, and it does not require energy. For instance, say you're cooking and you burn something by accident, and your house is real smoky. What do? You open the window. And why do you open the window? All right, because without even thinking about it, counterintuitively, that the smoke, where it's highly concentrated within your house, when you open up that window, it's going to diffuse to where it's low concentrated, which is outside the house, until essentially diffusion occurs until equilibrium is reached. But because outside is so big, outside is so big compared to your house, when equilibrium is reached, essentially not going to be any smoke in your house. Okay. Diffusion particles always move from where it's highly concentrated to where it's low concentrated naturally, and it does not require any energy. Okay. Now, sometimes, again, because the cell membrane has that hydrophobic core, so certain things that want to diffuse in or outside of the cell, they can't pass through the cell membrane, so they have what's called facilitated diffusion, which all it uses is a carrier channel protein, a carrier protein that facilitates it. So again, it helps, but it was still not requiring any energy. It just helped by giving a channel that it can go through the cell membrane. Okay, so facilitated diffusion just uses a channel or a carrier protein that allows whatever substance to move from the inside to the outside of the protein, or from the outside to the inside of the protein, still by diffusion, meaning still from where high concentration to low concentration. Okay, but you just need something to help you get through that hydrophobic core of the cell membrane. You have that carrier protein or that channel protein, so that's called facilitated diffusion. But remember, don't let facilitated confuse you. It has that diffusion attached to it. It still requires no energy. Okay, and then osmosis is just a specific type of diffusion. Osmosis is the diffusion of water. Okay, water also moves from high concentration to low concentration. Okay, but it's called osmosis. So you just get water diffusion from high to low concentration. We just give it a fancy name. We call it osmosis, but don't get confused. Osmosis, which is the diffusion of water from high to low concentration, is still diffusion, but in this case, it's water diffusing from high to low concentration, and we just call it osmosis. Give it a fancy name. And then the other type of passive transport is filtration. Okay, this type of transport that usually occurs across capillary walls. Okay, and again, you won't really talk about anything dealing with blood vessels or capillaries until anatomy 2. Okay. The science behind diffusion is really simple. So essentially, when something's highly concentrated, the collision between molecules in an area of high concentration is going to cause them to scatter into areas of less concentration. So that's very simple. Okay, so this difference is called a concentration gradient. So when you hear something going down, a concentration gradient going down, they're talking about going from high to low. When you hear something going up, a concentration gradient, you're going from low to high. And when you're going up a concentration gradient, going from low concentration of something to high, that does require energy. So that's going to be dealing with active transport, and we'll talk about that after we get through talking about passive transport. But going down from high to low, my concentration gradient does not require any energy. Okay, I'm going down a concentration gradient. And here's just a real simple experiment. A lot of times you can do. A lot of times you can just have two beakers filled with water, and one beaker is going to be really cold, and the other beaker is going to be really hot. Okay, the water inside the beaker is going to be really cold in one beaker, and the water inside the other beaker is going to be really hot. And what you do is you drop one drop of food coloring in both beakers, and you see how fast the food coloring disperses. Okay, so essentially diffusion is influenced by molecule size and temperature. So what happens with the cold water in the beaker, it's going to take longer, much longer, for essentially the food coloring to be evenly distributed in the water in the beaker. In the beaker that has the hot water in it, it's going to really quickly, the food colorant is going to be equally distributed in that water. So it's going to be looking like this really fast. Okay, but that's just a simple way to show you that diffusion is influenced by temperature and also the size of molecules. Okay, and remember, energy is not required. So again, when you go down a concentration gradient, meaning you're going from high concentration to a low concentration of a substance, going down the concentration gradient, no energy is required. And essentially, your plasma membranes, okay, create these concentration gradients by being selectively permeable. Okay, so they create these concentration gradients so things can become highly concentrated within the cell, okay, and then low outside the cell, or vice versa. They can be highly concentrated outside the cell and low inside of the cell. And essentially, those plasma membrane is creating these concentration gradients, and you're in these cells take advantage of the concentration gradients. We'll talk about that a little bit later also. So again, because the lipid core is hydrophobic, okay, molecules that can pass through are going to be lipid soluble, like certain fat-based hormones are lipid soluble, or anything non-polar, like certain gases, small gases that are non-polar, can pass through also. Okay, but larger molecules are going to need assistance by these carrier proteins, these integral proteins, these channel proteins are going to assist larger molecules go from inside to outside of the cell, or from outside of the cell to inside of the cell. Okay. So again, simple diffusion, your non-polar lipid soluble substances like gases, like oxygen, carbon dioxide, fat soluble vitamins, also certain lipid-based hormones can go right through by simple diffusion, pass right through the cell membrane. All right, so now when you get certain molecules that are larger, okay, and because they're larger or they might have a charge, they can't get through that hydrophobic core, they're going to need what's called facilitated diffusion. And don't let the name facilitated confuse you. All that means is that it's helping something out. It still does not require energy. Okay, so remember, no diffusion requires energy. Facilitated just means that it needs help. So it has these carrier mediated protein carriers or channel proteins that help move large polar or large charge molecules from either the inside of the cell to the out or the outside of the cell in. So you see right here, you have lipid insoluble solute, which may be sugar or amino acid. Okay, so essentially it cannot pass through this hydrophobic core of the cell membrane. So you have this carrier mediated, this carrier protein, which essentially the sugar or the amino acid binds to this part of the carrier protein. And then when it binds to this part of the carrier protein is going to change shape, and then it's going to release amino acid or the sugar inside the cell. Okay, so and understand that your cell has to be very, very organized. So these carrier proteins, these channel proteins, are very specific for certain substances that they carry. I'm doing a video recording. How was the movie?
It's good.
Yeah.
I mean, Tenet.
It looked like, just from the preview, it looked like that right amino acid binds to this carrier protein only after the right one binds. Then it's going to change shape, and then it's going to allow that amino acid, that sugar, to go within the cell. Okay, they're very specific for one substance. It mediated, facilitated diffusion. You still got that inward diffusion, no energy. You just getting help. In this case, you get channels. All right. That can transport molecules such as ions or water, all right, down their concentration gradient. Okay, so the hydrophobic core, so it goes through these channel proteins, and you have two types. You have leakage channel proteins, which are always open, and then you have gated channel proteins, which are controlled by chemical or electrical signal. Okay. And we'll talk more about the two types of channel protein and during the action potentials and the nervous system unit, because the leaky versus gated channels, okay, they play an important role in action potential. Okay. All right, so again, osmosis, just a fancy word for the diffusion of water from high to low concentration, okay? All right, and you use these specific water channels called aquaporins, okay? Aquaporins are specific channel proteins, okay? Still get some water diffusion through the hydrophobic core. All right, but the majority of the movement is going to be through these channel proteins, through these aquaporin channel proteins. Okay, but because water is so small, some of it can essentially sneak past the hydrophobic tails of the cell membrane. Okay. But again, the majority of it is going to go through the aquaporin channel proteins. Water is going to mostly go through that. Okay. No, get down. Which is the diffusion of water does not require any energy. No energy is required. Understand is that the particles, the solutes dissolved in the water, are going to affect the water concentration. Okay, so it's simple as that. So if you have low solute, that means that you're going to have high water concentration. Okay, low solute concentration, high water concentration, and the solute concentration and the water concentration are always going to be the opposite of each other. So when you have low solute concentration, it's going to have a high water concentration, and when you have a high solute concentration, meaning you have a lot of particles in this solution, you're going to have a low water concentration. Okay. All right. So there's a way that we haven't gotten to it just yet. So I believe it's well, when we talk about hypertonic and hypotonic solutions, all right, let me go back. I want to talk about that, but let me go back. Going on, and I'll share that with you. But just understand, so in this example right here, you have two solutions, okay? So you got two solutions that are separated by a membrane, okay? This is a membrane separating two solutions that is permeable to both solutes and the water. So it's permeable to both the particles and also in the water, okay? So you see this solution is more concentrated, okay, than this solution. This solution concentration is less. You have less particles in the solution in the water on this side, so the concentration is less. And this has more particles, therefore the concentration is greater, okay? So what happens when you have a freely permeable membrane? What happens is the water and the particles are going to move from high concentration to low concentration. So in this example, so remember, if you have low solute, you have high water. So this side has high water and low particles. This side has high particles and low water. But when you have this freely permeable membrane that allows water and particles to move, what's going to happen is the water on this side is going to move over here, and the particles on this side are going to move over here until equilibrium is reached. Okay, so diffusion always occurs until equilibrium is reached. And in this example, both solutions have the same osmolarity. Just think of it as they have the same concentration of solutes. Okay, they have the same concentration. Okay, so the diffusion is going to occur until equilibrium is reached. Okay, so again, it tells you the description up here. On the left, you have a solution with low osmolarity. You can think of it as low concentration of the solutes or low concentration of particles. The right has a solution with a greater osmolarity, has a greater concentration of solutes. Okay, so remember, low concentration of solutes, high water. High concentration of particles, low water. So just remember that. Now, in the next example, we're gonna have a membrane, a selectively permeable membrane that is only permeable to water, so the particles can't move across it. Okay, so when it's only permeable to water and the particles can't move across it, osmosis is going to occur until equilibrium is reached, meaning the water is going to move from where it's highly concentrated to where it's lowly concentrated until equilibrium is reached, meaning you have the same concentration of solutes and water molecules on both sides, okay, but you're going to have unequal volumes of water on both sides. So the volumes of water aren't going to be the same, but the concentration of the solutes are going to be the same, if that makes sense. Okay, the concentration is going to be the solute, because on the side that, say for instance, that has a lot of particles on one side of the membrane, if you have a lot of particles, you're going to need more water to dilute it down, essentially. Okay, that makes sense. So right here, you have your left compartment, which has very few particles. So low particles means it has a high concentration of water. Okay, just understand that. Low particles, low solutes, high concentration of water. So on the right side, you have high concentration of particles, low concentration of water, low concentration of water. So remember, this membrane, this selective permeable membrane, only allows water to move. The particles can't move, so only water move, and water is going to always move down its concentration gradient. It means it's always going to move from where it's highly concentrated to where it's low concentrated. Really the hardest part of this problem is figuring out which side of the membrane has the high concentration of water and which side has the low concentration of water. And again, you determine that by looking at the particles, the solid particles. If it has low solid particles like it does on the left, that means it has high water. Water is always the opposite of the particle. Okay, so low solute particles, high water concentration. Okay, high solute particles, low water concentration. So water go from high to low, from left to right. So you see the water is moving from left to right, and what happens is both solutions have identical osmolarity, meaning they are both the same concentration. Okay, they have the same concentration of particles. However, they have different volumes of water, because you needed more water on the right side because you have more particles on the right side, so that essentially would dilute the particles, so the concentration would be the same as it is on the left. Okay, if that makes sense. Microstatic osmotic. So also just understand that the movement of water causes pressure. So when you have a cell and you have the pressure of the water inside the cell pushing on the low water inside the cell, essentially. Okay, so that means you're going to create a greater tendency for the water to move inside the cell by osmosis, and that's called osmotic pressure. So osmotic pressure is the tendency of water to move into the cell. Okay, and we are going to stop here for the first, part 1 of chapter 3. We'll just take a look at the next slide. So the next lecture is going to pick up, and we will be talking about diffusion, but then we're going to get into isotonic, hypertonic, and hypotonic solutions. Okay.